So today's chapter is gene expression and epigenetics. It's a really very short lecture, shouldn't take long. This is all about how our environment is influencing the expression of genes. So again, we are going back to that nature versus nurture, and you will be, and of course, I think you've already learned about this in some of the previous lectures, that even genetic expression, how genes are influencing our traits, can be regulated by the environment. So, in 1945, Second World War was going on towards the end. Germany had blocked all entry points to some of the big cities in Netherlands, in Holland. So the Dutch people in these cities were starving. And then it was found out that at that time, the women who were pregnant, okay, because they didn't get enough food, so the fetuses there had prenatal starvation, right? And years later, it was found out that those children who suffered prenatal malnutrition when they grew up to be adults, compared to their siblings who had healthier gestations, who didn't face shortage of food, these children who faced shortage of food when they were not even born, before their birth, they had much higher risk of schizophrenia. And then a genetic analysis revealed, when they were comparing, when researchers were comparing what's wrong with with these people, because they face this starvation, and they found out that their DNA had more methylation patterns. That means there were more epigenetic changes. So DNA can be changed in two ways. Either you cause a direct change in the sequence of the DNA, right? So the DNA sequence is going like ATG, CC, CTGA, and there's a mutation, something happens, and the sequence changes. So that is a direct change to the DNA sequence. But another very important way in which DNA is changed is not by directly changing the sequence, rather by adding something to it or deleting something that was attached to the DNA. And most commonly, these are methyl groups, CH3 methyl groups, which when added to a DNA sequence can change the way the RNA transcription will occur. And if RNA is changed, then translation will be changed, then protein will be changed. And if protein is changed, then a trait is changed, right? So epigenetic changes in the DNA, and that is why it was because of the change in the methylation pattern. So when those children whose mothers starved, during that World War II, and because they were still and that was very early on in pregnancy, the very first few weeks of embryonic development, that's when a lot is happening, very dynamic time. And those fetuses, because their moms starved, so the fetuses were starved, and that caused changes in the methylation patterns of the DNA and made these kids, when they grew up as an adult, so prenatal malnutrition changed an adult phenotype, an adult trait. Okay, they were more likely to get schizophrenia. So changes in expression of genes may occur along, over time. Okay, a gene might be producing a protein A when it was at certain age, certain stage of your life, and then after a few years, that same gene expression could change. And instead of making a protein A, it is now making a protein B. And remember, whenever there's a difference in protein, this could mean a difference in the phenotype or the trait. And that same gene in one cell type could be producing protein A. Let's say that gene is in your heart cell, it is producing protein A. And that same gene, when it is in your nerve cell, in your brain, it is actually producing a protein B, not protein A. So depending on the location of the gene, depending on the time, okay, of expression, gene expression, that means phenotype, can change. And this may occur from microscopic to macroscopic level, from molecular level to organ level. A lot of these are epigenetic changes. That means chemical groups that can be added to, that can be associated with DNA, and these can be inherited, okay, passed on from one cell to its daughter cells. So we'll see some examples. Hemoglobin. Hemoglobin is a molecule that is present in your red blood cell. This is the most, you can say that when oxygen gets inside your body, hemoglobin is the one that gives oxygen a ride. So oxygen can travel throughout your body in blood. It can reach each and every cell. Every cell in your body needs oxygen. And that oxygen can be transported because of or thanks to this hemoglobin. Hemoglobin is present inside red blood cell. And red blood cells actually do not have a nucleus, they do not have DNA, they do not have any other metabolic machinery except for these hemoglobin. And hemoglobin has a complicated structure. It has 4 polypeptide, that means protein chains in adult hemoglobin. If you take any adult and you get the red blood cell out and you take the hemoglobin out, you will see that it looks like this. So there are two, you see there are 2 blue chains, 2 pink chains. We call them 2 beta chains and 2 alpha chains. And in the center of each of these chain is an iron molecule. We call it the heme group. This is where oxygen binds. Okay, so one hemoglobin molecule can actually bind with 4 oxygen molecules. One O2 here, another O2 there, another O2 here, another O2 here. So this is what adult hemoglobin looks like. 2 alpha chains, 2 beta chains. However, this hemoglobin molecule actually does not stay the same throughout the development of human beings. When after fertilization, when the zygote is made and then the zygote gets implanted on the wall of the uterus and then embryonic stage starts, okay, so from 2 to 4, 4 to 8, 8 to 16 cells are forming. At that stage, when hemoglobin is formed in the embryonic stage, instead of 2 alpha and 2 beta chains, it has 2 epsilon and 2 zeta chains. Okay, so that means the protein chains are different. After 8 weeks of development, when the embryo has become a fetus, then instead of these 2 chains, now it has 2 gamma and 2 alpha chains. And only in adulthood that you see, by 4 years of age, then the adult hemoglobin appears and it has 2 beta and 2 alpha chains. So the protein structure of the hemoglobin has changed. Okay, so the same gene which was coding for this protein at different stages of your life is expressing it differently. Now, if our blood plasma means the liquid part of blood. If you take it, and the plasma is 91% water, and there are some proteins, the most common proteins being albumin. Albumin is the most abundant protein in your plasma, fibrinogen, then globulins. So if you collect plasma from human samples and you analyze it for all the types of proteins that are present in plasma, you find about 40,000 proteins there. Okay, now conditions that are affecting the environmental conditions or even the stage of life can cause a change in this protein profile. Okay, you see here there were, where did it go? Maybe one of the later slides. Okay, that if you do a protein profile of the plasma and we show it in a pie chart, okay, or in a graph, whatever format, you will see that in the fetal stage, the protein profile looked so different from the adult stage. So in the fetal stage, the fetus might require enzyme A, B, C, at a much higher quantity than in the adult stage. In the adult stage, their work is done. So the adult age enzyme A, B, C might not even be there. So the protein profile has changed. Another example is that of the gland pancreas. So the pancreas gland is right here in the human body. You see, this is the esophagus, your food pipe going down. This is liver. This is the largest gland in the body. This is your stomach. And then from the stomach, you will see the small intestine coming out, makes a U-turn. And in that U-turn is this leaf-like pancreas. And the pancreas is known as a mixed gland or a dual gland. Why? Because it has both exocrine and endocrine parts. What's the difference between exocrine and endocrine? Endocrine glands make hormones, okay? Endocrine glands, they secrete whatever they're making, that means the hormones, directly into blood. While exocrine glands, they do not make hormones, they make other secretions, and then the secretion comes out through a duct, through a tube. That's the main difference. And usually a gland is either exclusively endocrine or exclusively exocrine. You might have heard of thyroid gland, right? A lot of people these days have trouble with the thyroid gland. The thyroid gland is an endocrine gland. It produces hormones, which are major metabolic players in our body. The sweat gland, it is an exocrine gland. It does not make hormone, it makes sweat. And the sweat comes out to the outer surface through a duct. Now this pancreas gland is very special because it has both exocrine and endocrine parts. The endocrine part makes 4 hormones, the 2 most important being insulin and glucagon. Insulin is that hormone that brings the level of glucose down, right? So people with diabetes, usually if they're not making enough insulin because the pancreas is not working, then they have to inject themselves with insulin. While another hormone is the opposite of insulin called glucagon, which actually brings the level of sugar up, glucose up in your body. So that is the job of the endocrine part of pancreas, hormone secretion. What does the exocrine part do? The exocrine part makes digestive juices and enzymes because it is helping in the digestive system. As food is passing from the stomach into the small intestine, it has to be digested and the nutrients have to be absorbed. So those are the two main jobs. Now, how does the same gland end up having an exocrine and endocrine part? It's because of the differential gene expression, okay? Depending on whether that gene will get expressed or not, in which part of pancreas, then that part would develop as exocrine or endocrine. And the gene here is a transcription factor. We call it the PDX1. So if the PDX1 is activated, then those cells would follow the exocrine pathway. They will go into, and develop into exocrine part of the pancreas gland. While those cells that do not get this activation of PDX1, okay, and they're responding to other genes, they would actually make the endocrine part. So you see, this is the stem cell. The stem cell is differentiating into progenitor cells. This progenitor cell gets the PDX1 gene activated, so it would make the exocrine part, the acinar part, while this progenitor cell would not get the PDX1 activated, and it ends up making these 4 different cell types, which make 4 different types of hormones in the pancreas. So the expression of genes, whenever it's gene expression, what helps us in learning about the gene expression? It is the making of the proteins. So proteomics, and we have learned this definition in one of the earlier lectures, is the study of collection of all proteins made by your genes in cells, tissues, glands, and the entire body, okay? And we have actually millions of different types of tissues, of proteins made in our tissues. So if you broadly classify them, there are 14 categories of proteins, including the immunoglobulins. The immunoglobulins means the antibodies, your soldiers. So you see here, our proteomes change over time. So this is the pie chart, and this shows what was your proteome in from conception to birth stage, while this is later on, okay? This is through old age. And you see here this enzyme part, when it was in the embryonic stage, you see how much was, only this much part was made up of enzymes, while if you come here, all of this is enzymes. The transcription factors here had a much bigger share. The transcription factors here have a smaller share. So your proteome, your protein profile in your body is changing with time as you are getting older. So what controls these gene expressions? We keep saying that gene expression changes because in response to environmental triggers or something else. Yes, there are a lot of regulators. And actually, if you look at a human genome, very few, very little part of the human genome is actually making proteins. So what is the rest of the human genome doing? The rest of the human genome is controlling the protein coding part of the genome. And we have different types of regulators about which we still do not know everything. So much is still a big mystery, and that was a big disappointment when the human genome was decoded, because people had expected a lot more, more than the 23,000, 24,000 protein encoding genes. People had expected a lot more, but it was found out that, more significant part of the human genome did not code for proteins. It was mainly regulatory regions and regions of unknown function. So a protein encoding genes would have some control over its expression level, right? There is a promoter sequence, that means a sequence that comes before the gene, which controls the expression of the gene, okay? And much of this control of gene expression can occur in these 2 ways. There are other ways also, but these are the 2 main ways. The first one is called the chromatin remodeling. Chromatin remodeling acts like an on and off switch for a gene, okay? So this gene can code for a protein, but will that gene make the protein now? That's a big question. So you have to turn, is that gene turned on? Then the gene will make the protein. If the gene is turned off, it is capable of making a protein, but it will not make a protein. Or it will not make a protein in this tissue because it stays off in this particular tissue. MicroRNAs, these are hot topics, hot research topics these days. A lot of research is being carried out on the role of microRNAs in cancers. These act like dimmer switch. So although the gene is expressing what about the severity of the trait, okay, this would act like a dimmer switch on top of that chromatin remodeling. So how does chromatin remodeling occur? We are trying to understand it here in a very, very simplified manner. If you remember the structure of your genetic material, the chromatin threads, it includes DNA and protein, right? So DNA is the thread-like material, and there are histone proteins that bead on a string arrangement. So as the DNA is wrapped around those, rounded ball-like structures, those are the histones. The histones play a major role in gene expression. Why? Because which part of the DNA is winding around the histone and which part is free, that is decided by the histone. So the histones would expose the DNA when and where it is to be transcribed, and they mask that part of the DNA when it is not to be transcribed. Okay, so in this tissue, okay, this part of the DNA has to be exposed. So the histones move slightly. They unmask that DNA. In this tissue, it does not have to be, or at this stage, it does not have to be expressed. So they shield the DNA. So this role is being played by the histone proteins. And then there are the small molecules that can bind to the histones and cause epigenetic changes. We've been talking about epigenetic changes. These groups are the methyl groups, the acetyl groups, and the phosphate groups. So these groups can add to DNA and cause changes. How? So let's say this gene had a transcription off. What can cause the transcription to turn on? That means from DNA to RNA. What will cause the DNA to make RNA? If epigenetic changes occur, if you add an acetyl group, it will turn the expression of the gene on. This is like an on switch. If you add a phosphate group, it turns transcription on. If you delete a methyl group from here, it turns transcription on. So deletion of methyl group, but addition of acetyl and phosphate group cause transcription to start. MicroRNAs, micro means really small, RNA molecules, right? These are special type of RNAs in general, which are called non-coding RNAs. These RNAs do not make proteins, okay? These are really small, only 21 to 22 bases long, okay? And they are responsible for controlling at least 1/3 of the protein encoding genes. So how does a microRNA work? Or usually, not just microRNA, there are also siRNAs, small interfering RNAs. So the microRNA can go and bind to an mRNA. As it binds to an mRNA, now the mRNA will not go and bind to the ribosome to start making proteins. Translation will not occur. So if you think of this one is the DNA, right? The transcription factors. So this long green mRNA has been transcribed. Now this mRNA should go out from the nucleus into the cytoplasm, and it should now bind to the ribosome, and then it would ask the tRNAs to come and protein synthesis would start. But as this mRNA moves into the cytoplasm, these small microRNAs, which are complementary to some regions of the mRNA, they come and bind to their complementary regions. And now the mRNA is not free to move around and look for the ribosome or call the tRNA. So protein translation has been stopped. Okay, especially in cancer research, it has been found that people have different microRNA profiles. When it was compared, when comparison was done between cancer patients and normal healthy controls in different types of cancers, not restricted to any specific type of cancer. And they, the controls versus the cases, they were found to have different microRNA profiles. So certain microRNAs are more or less common in cancer patients than in healthy ones. And a technology is very commonly used in molecular biology. It's called RNA interference, RNAi, where we use small interfering RNAs, siRNAs. For example, I work on prostate cancer, right? And there's a particular gene of interest that I work on, which is called P75 in short. So this P75 gene has been found to be overexpressed in prostate cancer patients. That means this gene really has a very high expression in patients who get prostate cancer, and this protein has low expression in healthy individuals, in controls. And when this protein has very high expression, when this gene gets very high expression and makes a lot of P75 protein, the patients become resistant to chemotherapy. right? So a part of my research was to try to see if I could somehow bring the level of this protein down in the cancer cells. Can I make these cells more responsive to chemotherapy? So what we did was we used this technique of RNA interference, and we designed small interfering RNA molecules, siRNAs, that were complementary to the RNA of my gene of interest called the P75. So these were commercially made. We ordered it to a company. The company supplied it to us. And first we had to confirm that these were actually blocking the gene. So, we have cancer cells growing in the labs, and we treated them, and we wanted to see with different, like gel electrophoresis, we wanted to see if the expression was down. And we saw that, yes, the siRNAs were working. They were going and binding to the mRNA and not letting the mRNA get translated. So the P75 expression was coming down. Then the next part of my experiment was to treat these cells with chemotherapeutic drug, docetaxel. That is the last word in prostate cancer. This is the last chemotherapeutic drug that is used to control the spread of cancer. So then I started treating my cells with docetaxel, and I wanted to see if the cells were now responding better to the chemotherapy. So I had, on one hand, I had cells with the RNA knocked down, okay? And on the other hand, I had cells which were not knocked down. That means they had high P75 expression. And we saw that when we knocked down this gene, the cells became more responsive to chemotherapy. That means we were sensitizing these cells to chemotherapy, and this was a good result, a positive finding. Now, maximizing genetic information. This means that from the 20,325 genes on our human genome that have been found to be responsible for making proteins, how do we end up making millions of proteins? Just from those 20,000 genes, we are making, so many proteins. How are we maximizing these genetic information? So this maximization occurs at different levels, at different steps, okay? Because from these genes, from these DNA sequences, they encode about 100,000 mRNAs. Okay? Because remember, from DNA to RNA, we have those exons and introns, and the introns would get spliced out, and then the poly A tail is added, everything, a lot of processing occurs. And then those 100,000 mRNAs are used in different combinations, and they're processed in different ways, modified in different ways to make more than 1 million proteins. How does that occur? There are so many modifications, post-translational modifications. If you see this figure here, this is the central dogma. If you remember, this is a cell with the pink cytoplasm. The blue part is the nucleus. This spiral structure is your DNA, double helix. So what is this? The central dogma, that DNA can replicate itself to make more DNA. This is replication. Then from DNA to RNA is transcription. Then from RNA, mRNA comes out, makes protein. This is translation. So replication, transcription, translation, right? In the DNA, there are about 20,325 genes. RNA, about 100,000 mRNAs, and then 1 million proteins. How is this maximization effect occurring? This is because of all of these modifications which are listed on this side. From DNA to RNA, there could be chromatin remodeling. Which genes are turned on, which genes are turned off? Depending on that, the mRNA will be differentially made. Again, is this stage when the precursor mRNA has been made from the immature mRNA to a mature mRNA, there could be alternate splicing. Which introns just got deleted? Okay, was an exon also deleted? Up to which exon was the splicing done? And there could be a microRNA that could come and sit down on the RNA and stop the translation. So all of this can occur at the RNA stage. Then from RNA to protein translation, when a protein is initially made, it is not functional, right? That's the primary protein. In order for the protein to become functional, it has to fold in complicated manner. It has to go from primary to secondary to tertiary to quaternary structure. And that protein folding could be different. That same chain of polypeptides, let's say that chain had A, B, C, D, E, F, up to F amino acids. That chain could make a loop, that chain could make a whorl, the chain could make an arch, depending on how the chain has been folded. Or were some polypeptides shortened? Were some just cut, you know, to make, to live? This one chain of A, B, C, D, E, F, G was probably cut in the middle, and it came down to A, B, C, and another chain from D through F. Was there some sugars added that would result in the making of a glycoprotein, or some polypeptides would come together? So all of these are strategies by which our body maximizes genetic information. From that same DNA, we end up making a variety of proteins because of all of these changes that can occur at different stages. For example, this shows you how at the level of protein, depending on how the polypeptide chain is folded or how it is cut, it can give rise to a disorder, or the trait is changed, okay? So one gene can encode more than one protein, and then if that protein can be cut to form two different proteins, and this is a disease called dentinogenesis imperfecta. This is autosomal dominant. What are the symptoms? There is tooth developmental disorder. You see these brownish, discolored, weak teeth. These teeth are really brittle. The dentin, okay, the part inside that makes the teeth, is very weak. And this is because these are made by the same gene. You see here the DSPP gene. The DSPP gene, after transcription, makes a DSPP mRNA, and this DSPP mRNA should stay like this, DSPP precursor protein. But in some cases, it gets cut into two. And both of these are then responsible, you know, both of these are responsible for making this abnormal condition, because the protein got cut. So there was an abnormality that was introduced. Now remember, what we've been talking so far is most of the human genome is not coding for protein, right? Most of the human genome is actually controlling this small part of the genome. So only 1.5% of your DNA is making protein. The rest of the genome is a lot of non-coding RNAs. Okay, there are introns there, that the DNA is made up of exon, intron, exon, intron. There are promoters and regulatory regions. There is also surprisingly viral DNA. How this happened? This happened during the course of evolution. About 8% of our genome is actually derived from RNA viruses. RNA viruses are known as retroviruses, okay? And this is evidence of evolution, how, you know, during the course of evolution, our ancestors were infected by viruses. And because viruses have a very strong tendency to go inside the nucleus and to integrate with the genome of the host cell. So you see here, this virus might have got inside at some point, you know, in evolution, in one of the ancestor cell, and that cell, if it was a sex cell, then that information was passed on to the next generation, and a little bit of the RNA from the virus got integrated. You see those red dots with the human genome. And then over the course of time, because these were not lethal, these were not killing the human beings, or maybe not giving rise to a huge problem or a big disorder, so it just stayed there, okay? It was retained. It was not kicked out. A lot of the times, and you will learn about these things in subsequent lectures, sometimes if a mutation is really detrimental, then those individuals, nature makes them sterile. Those individuals cannot reproduce, and that is how nature ends that mutation in that particular individual, so that that particular mutation cannot be passed on to the next generation. But these mutations were not that detrimental, so over the course of time, they stayed, and not only stayed, they increased. You see how the red dots have increased. So this is how viral genome came to be incorporated into human genome. The non-coding RNAs, okay, the non-coding RNAs, if you go to any seminar these days, big conferences, cancer, neuroscience, you will hear a lot of talks on non-coding RNAs, because maybe 2 decades ago or a decade ago, people were not aware of these, you know, the work of these, and they were mainly focused on only protein encoding genes, because they thought that is the most important part of our DNA, that is the one that's making DNA, making protein. But now people know that these may be even more important than the protein encoding regions, because they are controlling the protein encoding regions. So the non-coding RNAs means any RNA that does not code for a protein. So it would include the ribosomal RNAs and the tRNAs, okay? And usually these RNAs are transcribed from what we call as pseudogenes. Pseudo means fake. So these are not protein encoding genes, they are pseudo genes, and these RNAs are there for other purposes, not for making proteins. So they will never get translated into protein. We also have in our genome repeats, okay? And repeats have also confused scientists for a long time. Some of the common types of repeats are called transposons. Transposons, the scientist who discovered this, she received a Nobel Prize in physiology or medicine for her discovery. So transposons are sequences that are mobile, okay, that jump about the genome. So these are two types of transposition, two types of jumping around of DNA sequences. Imagine this is a strand of DNA. This part is the transposon that just likes to move around. So one type of movement can be a cut and paste movement. Now this DNA wants to go there. It will simply cut itself, just like we do in Word document. We cut it and it gets pasted here. Okay. So this DNA sequence has been interrupted. A big change has been brought in the protein expression because the DNA sequence has been changed. Another mechanism of transposition is copy and paste. This is the transposon, and it doesn't cut and paste. Instead, it makes a copy of itself and it pastes there. So this one stays as usual, but here again, the DNA sequence has been interrupted. Okay, we see a lot of repeats in Y chromosomes. We see a lot of repeats. Every chromosome towards the end telomeres has repeats. Now, these are some non-protein encoding parts of the human genome. We talked about viral DNA. We talked about non-coding RNAs. You already know about what is tRNA, what is ribosomal RNA, pseudogenes. What are introns? Those are regions of the DNA that do not make RNA. We also learned about transposons. We've learned about telomeres. Remember, telomeres, shortening of telomeres signal to that chromosome that you have become old and senescent and that you're not going to divide anymore. And this is what in cancer cells goes wrong. There's an enzyme called telomerase that makes the telomeres immortal, keeps them, prevents them from getting shorter. So the cells just keep dividing and living forever. And we are done. Any questions? Okay, so then you don't have discussion session today. Your discussion is tomorrow. If you have any question other than what we learned today from any other chapter also, you're welcome. No? So we'll end the meeting today. I'll see you tomorrow.